So what we're going to do now is I'm just going to go and talk really briefly about osmosis and diffusion again. And there is going to be, if I can, yeah, I'll show you. There's going to be a pre-lab for the osmometer lab, but the pre-lab is really easy. If you understand what the concepts are, it's not going to take you more than like 10 minutes. But it's important that you understand the concepts. So that's why I want to just briefly go over the osmometer lab. So we should just make sure everybody's on the same page as what's osmosis.
It's like the movement of water across a membrane from a high concentration to low concentration.
So diffusion of water, and we usually say across a semi-permeable membrane. Normally when we think of diffusion, we think of some solute, some dissolved thing moving from one side to the other. In osmosis, we have some kind of membrane that allows water to move back and forth. H2O can freely move back and forth, but some solute is trapped. So, and again, it could be sugar or salt or whatever. So the solute cannot move across. So that's why we say semi-permeable membrane. Water can freely go back and forth. This solute though is trapped. And therefore, rather than thinking about concentration of solute driving the motion of a solute, instead, you kind of flip it in this weird way of like concentration of water. If this is side A and this is side B, in this picture that I've drawn here with the solute on, dissolved there on side A, which way is the water going to move? Is it going to move towards side A or towards side B?
Side A.
It'll move towards side A because again, there's two ways to think about this. One way is the concentration of water, which I find really awkward, but I'll say it anyway, just in case it is useful for people. You can think about, okay, there's more water on this side than this side, because if there's dissolved stuff on side A, that means there's not as much water there because some of this stuff is solute rather than water. So that means there's a higher concentration of water here. So water is going to move from high to low concentration of water. Like, to me, that's just kind of weird and awkward to think about concentration of water, because usually you think of a concentration of something in water. So when I think of osmosis, I usually think about kind of the salt drawing in, it's like kind of, the water, is kind of drawn, to the higher solute concentration. Again, when I think about it, I am not proud of this whatsoever, but me, as well as probably many of you, when you're kids, there is something kind of crazy cool about putting salt on a slug and watching it get all shriveled up. And now I feel really bad about it as a grown-up, but as a kid, I did it. And it's like, what's going on? How come this slug shriveled up when you put salt on them? It's osmosis. The water in this little dude gets pulled out from the more watery slug to the less watery salt. It's osmosis. So for me, it's just kind of natural to think about the saltiness drawing up the water going to where it's more salty. This is why we preserve foods by salting them or by candying them. If you have a really high concentration of sugar or salt, some little random bacteria or yeast or whatever lands on there, it's not going to grow. It's just going to shrivel up. So you can have some, candied ginger and it's never going to rot because any bacteria that lands there is just going to get desiccated. So whatever works for you, again, like I said, for me, when I think of osmosis, I think about the water moving towards the high concentration of solute. But that's just me. The thing that's important to remember in osmosis, as in any kind of diffusion, it's the difference between the two sides that drives the motion. So rate of osmosis driven by what we call the concentration gradient. Difference between the two sides. Right, so I could have some membrane here, and I could have both sides super concentrated. This could be 40% sucrose, like, really super sugary water. This side could be 40% sucrose, super sugary water. Which way is osmosis going to happen?
Both or neither?
Neither. There's not going to be any net movement of water because there's no gradient. There's no difference between the two sides. So it's important to remember when you're thinking about a rate of osmosis, how fast is the water moving? It's not just about what is the concentration of solutes. It's about what's the difference between the two sides of the membrane. That's going to be critical in coming up with your hypotheses today, or for Thursday, I should say. You always want to think about the rate of osmosis, how fast that movement is happening. It's driven by the gradient, by the difference. And in our experiment, we're going to make an osmometer, measure osmosis. It's going to be simple. It's basically going to be a glass tube. We're going to have what we call dialysis membrane. Membrane lets water through, lets water pass, not sucrose. We're going to put it in a beaker with water. This is just going to be distilled water. And then inside this little bag, in this membrane bag, I'm going to add in different kinds of sugar water. One case is going to be 20% sucrose. One case is going to be 40% sucrose. What's going to happen as soon as I let this thing start to go? I have this little membrane that lets water cross, but not sucrose. And this little membrane bag is filled with sugar water, lots of sugar in there. It's distilled water, nothing dissolved in this bathing solution. What's going to happen?
The water's going to move towards the bag.
Yeah, the water's going to
Of the
move into the
40%?
bag. Right? Remember, it's like putting salt on a slug, right? The water is going to move for where there's no solute. It's like zero osmolarity to where there's lots of stuff dissolved in the bag. So our water is going to move in. And as it moves in, we're going to start seeing the solution start to move kind of up the tube. And we'll be timing it. We're gonna look at like 5 minutes, at 10 minutes, at 15 minutes, so we can see the rate. This is gonna be our measurement of the osmotic rate by seeing how fast is the water going up the tube as the water enters across the membrane. So this is the osmometer. So now let's get a little trickier. What's our independent variable and our dependent variable?
Your independent variable is your solute concentration gradient, and your dependent is the rate of osmosis.
So exactly. And notice that she didn't say solute concentration, right? Even though we have different solutes in there, 20% versus 40% sucrose, what's really relevant here is not just the concentration, but it's the gradient. It's what is the difference between the two sides of the membrane, 0 to 40 versus 0 to 20, right? Because I could have, if I had 20 outside, this would be nothing and this would be pretty fast. It matters what the gradient is. So this independent variable, we use different solute concentration gradients across the membrane. Dependent variable, we'll be able to look at the rate. How fast is it going up the tube as time goes by? What do you expect to find? What do you think the relationship is going to be?
The higher the concentration gradient, the faster the rate.
Yeah, exactly. Right, because this is just a special version of diffusion, and that's what we're talking about in lecture with diffusion and facilitated diffusion. Just, there's lots of things. In fact, rate of diffusion, we'll talk about this in more detail. There's this whole thing, Fick's law, but it's proportional to a variety of things. This means proportional. One of the main things it's proportional to is the concentration gradient. Gradient, what am I writing? So the concentration gradient is going to be a major thing that affects the rate of diffusion. So if we have a larger concentration gradient across the membrane, all else being equal, we're going to expect a faster rate of osmosis. What other things affects rate of diffusion?
Permeability of the membrane.
Yeah, the permeability of the membrane. The easier it is for the water to move across, that's going to make it faster. What other things?
Temperature of the water.
The temperature, yeah, totally. The higher the temperature, we talked about diffusion is just driven by that native energy and motion of the molecules. So higher temperature means faster rate of diffusion. What other things affect rate of diffusion?
Enzymes?
Those aren't going to do it, right? Enzymes are going to speed up chemical reactions, but they don't really have a role to play here in diffusion. Diffusion is this kind of random walk of molecules kind of floating around.
What about surface area?
Surface area, another huge one. The higher the surface area, the more you're going to be able to cross in a certain amount of time. This is why you've got all that crazy microvilli unfolding in your your small intestine to maximally absorb your nutrients, or in the proximal convoluted tubules of your nephrons to reabsorb things back into the body from the filtrate and all that. So surface area is going to affect it, permeability, temperature, concentration gradient. We'll see. Go on.
Will the type of solute have an effect?
So it will. In rates of diffusion, it's going to be like molecular weight will be kind of negatively correlated, because the larger a molecule is, the heavier it is, the kind of slower or more sluggish it is. The same amount of energy, like if I have, you know, right, I can have, let me stop this for a second. If I have like a finger flick of energy with a smaller thing, you just saw it go sailing. If I take a finger flick of energy with this much more massive thing, it barely moved, right? So the same amount of energy for a large thing, it's not going to move as far and fast as the same amount of energy with a little thing that's going to be zipping along a lot more. You know, so we're going to look at that in more detail, but just for your pre-lab, there's no pre-lab for the demonstrations thing. The only pre-lab is for the osmometer lab. And it's just what we just talked about. It's basically, we saw the independent variable. You're going to be looking at different values of the gradient across that membrane in the osmometer. We'll have a lower one, like 20, 0 versus 20 versus 0 versus 40. And then the dependent variable is just going to be, how does that affect the rate of osmosis as measured by how fast the water's going up the little tube? So for Thursday, you know, it should just be objective, hypothesis, source of variability. Again, at this point, you're getting good at that. It shouldn't take you more than 10 minutes, I'd say. And plus you've got new mad skills in how to upload your PDFs directly into Canvas. So that's going to be even easier than it ever was, probably. You know, and in terms of the analysis of the jumping stuff, if you have questions or you're feeling like overwhelmed, like, holy crap, you know, I'm going to be here. I'll help guide you through this stuff. There's also lots of resources there. So, and you've got a couple of weeks, so it's not like you got to do it tonight. You have time to kind of, and if there are skills you're not as comfortable with now, I promise you, you'll actually like knowing them because you'll use them in other contexts in your life. That's something I never feel guilty about, getting people getting better on Excel, because you just go on a vacation and it's going to help you figure out who owes what when you split up the finances or whatever. Excel is super useful. Or you're planning a party and you're trying to keep it under budget, you know, Excel is your ally.